Hello everyone, I'll be with you for microbiology. Microbiology, although tough, but it is very interesting, and once you know it, you will really like the subject, and infectious diseases is linked to it. I would be talking and integrating it with infectious diseases so that it is easy to remember. But the basics will be taught first. And here we go with the cytoplasmic structures, nucleus, chromosomes, etc. about the eukaryote and the prokaryote. Now, the eukaryote has nuclear membrane. It is has linear chromosome. Ribosome is ATS and cell membrane contains sterols. They have mitochondria as well as Golgi bodies present. The respiration is via mitochondria, whereas in a prokaryote, nuclear membrane is not there. They are circular, haploid DNA. Ribosomes are 70S. Cell membrane has no sterols, there is no mitochondria, no Golgi bodies, and no endoplasmic reticulum. And the respiration in a prokaryote is via cell membranes. You have to remember this table. I have made it very easy for you to remember. Now, microbiology includes viruses, bacteria, fungi and parasites. Viruses are very minute. They are 0.02 to 3.3 microns. Bacteria are 0.3 to 2 microns. Fungi 3 to 10 and parasites are 15 to 20 microns. Now, the cell type, viruses are acellular. Bacteria are prokaryotic cells, whereas fungi and parasites are eukaryotic. Viruses, they have either DNA or RNA. Bacteria have DNA and RNA, one chromosome, and there are no histones here. Whereas fungi and parasites have DNA and RNA. Viruses replicate in the host cells. They have polycistronic messenger RNA and post-translational cleavage, whereas bacteria are mono or polycistronic messenger RNA. They have that, and the DNA replicates continuously. In the parasites, they have two phases, G and S phases. They have monocistronic RNAs. Viruses, they use the host organelles and they are obligate intracellular parasites, whereas for the bacteria there is no membrane bound organelle. In fungi and parasites, mitochondria and other membrane bound organelles are there. Viruses have no ribosomes. Bacteria have 70S ribosomes, 30S plus 50S, and fungi and parasites, being eukaryotic cells, have 80S ribosomes, that is 40S plus 60S. We should know about the cell wall, cell membrane also. Viruses have no cell wall, but they can have an envelope which has no membrane function. And these viruses, they make and assemble the viral components by replication, whereas bacteria has asexual replication by binary fission and membranes have no sterols. The only bacteria having sterols is Mycoplasma. This is the MCQ asked, which have cholesterol. And the cell wall is made up of peptidoglycan, whereas in fungi and parasites, the membrane has ergosterol as a major sterol, and this is from ergosterol is present in fungi, whereas parasites, they have cholesterol. Fungi, they have complex carbohydrate cell walls of chitin and glucans. Again, the question asked. The question goes like this, that they can ask about the cell wall of fungi. That what are the components? The answer is usually chitin, chitin, whatever you call, and glucans. Parasites have no cell wall. Now, this is the morphology of a bacteria which you see here. We have pili, we have a capsule here, okay, and then we have flagella, ribosomes. It has nucleoid and also plasma membrane, as you can see. This is just a basic structure of bacteria. Then we have the important normal flora. This is a very important slide. Normal important flora. You can see that the sites, I will discuss it with the sites. Just a minute. So, this is blood and internal organs. Now, the most common and medically important organisms are none. Remember that blood and internal organs are sterile. Even urine is sterile. The question can be asked. Now, the cutaneous surfaces, they usually have Staphylococcus epidermidis on them. Less common are Staphylococcus aureus, diphtheroids, streptococci, and some anaerobes like peptostreptococci and yeasts. In the nose, it is the Staphylococcus aureus which is usually present, but Staph epidermidis, diphtheroids, and streptococci can be present. But you just remember the nose has Staphylococcus aureus. In the oropharynx, we have viridans streptococci. Less common are non-pathogenic Neisseria, which are non-typable, H. influenzae, and Candida. Gingival crevices, they have anaerobes, which can be bacteroids, Privatola, Fusobacterium, Actinomyces, Streptococcus, etc. Stomach also has no bacteria. Remember that the pH of the stomach is 2 and that kills the bacteria. Then colon. Colon has, in the babies who are breastfed only, the bacteria that are present are Bifidobacterium. The adult has bacteroids, Privatella, Escherichia, and Bifidobacterium. Less common can be Lactobacillus, Enterococcus faecalis, etc. Vaginal. Vagina has Lactobacillus. This is again a question asked that the normal bacterial flora of the vagina mainly has Lactobacillus, although Streptococci can be present. The importance of Streptococci in vagina is important. We will discuss it subsequently. Gram-negative rods can also be present in vagina. Diphtherias, yeasts, Villanella are all present. But remember, it is usually the lactobacillus that is present in the vagina. Now, morphology. We should know that bacteria can be gram-positive or gram-negative. And all gram-positive are cocci or bacillus. Gram-positive cocci are streptococcus and staphylococcus, whereas gram-negative cocci is Neisseria. Gram-positive rods are Corynebacterium, Listeria, Bacillus, and Clostridium, whereas gram-negative are Escherichia, Shigella, Salmonella, Yersinia, Klebsiella, Proteus, Vibrio, Campylobacter, Helicobacter, etc., to name a few. So you should know that which organism is gram-positive and which is gram-negative. That will help you in certain diagnosis. As we go subsequently, you will understand it. The spiral. Spiral bacteria are spirochetes, which are Trypanoma, Borrelia, and Leptospira. Remember this. Now, branching filamentous growth is seen with actinomyces, which are anaerobic, and the partially acid-fast nocardia. The bacteria which are pleomorphic are Chlamydia and Rickettsia. Mycoplasma has no cell wall. Now, you should know the differences between the gram-positive and gram-negative organisms. Gram-positive cells have two layers inner cytoplasmic membrane and outer thick peptidoglycan layer. Remember this. They have low lipid content and no endotoxin. The only gram-positive which has endotoxin are Listeria monocytogenes. There is no periplasmic space, no pore channel, and they are vulnerable to lysozyme and penicillin attacks. Whereas gram-negative cells, they have three layers. The outer membrane is the LPS, the inner membrane is the cytoplasmic membrane, and the peptidoglycan layer is thin. Remember, in gram-negative cells, there is thin peptidoglycan layer, whereas in gram-positive cells, the peptidoglycan layer is thick and outer, whereas here the outer layer is LPS. Gram-negative cells or gram-negative organisms, bacteria, they have a high lipid content. This is this LPS which is actually endotoxin and has lipid A. They have periplasmic space, they have pore channels, and they are resistant to lysozymes and penicillin attacks. Now, you can see these cross linkages here, and here I will be showing you the main differences between the gram-positive cell wall and the gram-negative cell wall. Now, this is the plasma membrane I talked about. There is a periplasmic space. Then this is the outer membrane. Okay, and this is the LPS, that is lipopolysaccharide. Whereas here in gram-positive organisms, you can see the plasma membrane. There is no periplasmic space, and this is the peptidoglycan. Now we move towards the next slide where I will be talking about gram stain. These are the gram positive and this is the gram negative organism staining. In the gram positive organism, first fixation is done. Fixation is done by drying, natural air drying, or just we put the slide over the flame to fix the smear on the slide. Then we add crystal violet. After adding crystal violet, you can see that both the cells, gram positive and the gram negative, they stain purple. After that, we have to give the iodine treatment, and after the iodine treatment, what we do is the decolorization. When we do the decolorization, in the gram positive bacteria, decolorization doesn't occur, whereas in the gram negative bacteria, as the cell walls are very thin, very easy to decolorize them, and then after decolorization, counter stain safranin is added and safranin will stain pink. So gram negative will appear pink, whereas gram positive will appear purple under the microscope. So you should remember the steps of the gram stain: fixation, crystal violet, iodine treatment, decolorization, and counter stain by safranin. Medically important bacteria are the following. Some bacteria have rigid cell walls, whereas others lack cell walls. The bacteria which lack cell walls are Mycoplasma and Ureaplasma, whereas the bacteria with rigid cell walls can be simply unicellular or filamentous. Remember, filamentous bacteria are Actinomyces, Mycobacterium and Nocardia. Okay, and the flexible ones are Borrelia, Leptospira and Trypanosoma. And here we have obligate intracellular parasites which can be Chlamydia, Coxiella, Ehrlichia and Rickettsia. These are the free living bacteria. Free living bacteria are gram positive or gram negative. I've already talked about them, that they can be cocci or rods. Cocci are gram positive, cocci are Enterococcus. Remember, this Peptostreptococcus is an anaerobe. Staphylococcus, Streptococcus, whereas Rods can be Bacillus, Clostridium, Corynebacterium, Lactobacilli, Listeria, or Propionibacterium. Gram-negative can be cocci, non-enteric rods or enteric rods. Remember, enteric rods means that they are present in the small intestine, and those which are present are mainly, you should remember, Escherichia, Helicobacter, Klebsiella, Proteus, Salmonella, Pseudomonas, and non-enteric rods are Legionella, Haemophilus to name a few, Bartonella, Bardetella, Brucella, etc. Even remember, Pseudomonas can be enteric as well as non-enteric. It can cause respiratory infections also and can cause GI infections also.